The monomers of a protein are amino acids. So proteins are a polymer, the monomers are amino acids, and they have a backbone that is always going to look like this. There's a central carbon, there's a amino group on one side, one of those functional groups, and there's a carboxyl group on the other side. And then the R group varies from amino acid to amino acid. So this is often referred to as the backbone or the side chain. Okay? There's a total of 20 amino acids, 20 different kinds, and the R group is what varies between them. And so, for example, some R groups are polar, some are bases, some are acids. They're all different, okay? And that side group is what helps to determine also the shape of the protein. So to give you some examples of some of the properties of these R groups, these side chains, some of them have side chains with lots of carbons and hydrogens stuck together, so they're non-polar, right? That means they won't mix with water. Some of them are in a ring, and that ring is basically just carbons and hydrogens, so these are known as aromatic amino acids, and they are also non-polar. Meanwhile, some of these have R groups that have oxygen, and we know they share electrons unequally. That makes them polar. Or they have R groups with acids or bases. You'll know that because they'll draw them as having a positive or negative charge. These do interact with water. These are hydrophilic. So of course these groups up here are hydrophobic. These examples are hydrophilic. And once again, I stress that students don't always take this to heart, but I want to know why O and H are hydrophilic. Why? And you'll have to walk me through that because of electronegativity, unequal sharing of electrons, so on and so forth. The water molecule is a polar molecule, therefore, You want to be able to explain all that. I'm not going to explain it all again right now. I want you to work on it yourself. Okay, so here are some examples of side chains that are hydrophobic, not liking water. Notice the backbones are in purple here. We ignore those. Don't pay attention to the backbones. So don't pay attention to the amino group and the carboxyl group part. But look here, lots of C's and H's. We know that those are non-polar covalent bonds because C's and H's have the same electronegativity. Therefore, those would not like water. So in a protein, they would turn away from water. Same goes here. And here's one of those examples of an aromatic chain, aromatic ring. If you were able to see the C's and H's here, this is a bunch of C's and H's stuck together. So those are hydrophobic. Hydrophilic examples are shown here, where we see an OH, for example. We know that when you have an O stuck to an H, the O is a little bit negative and the hydrogen's a little bit positive. Okay. Here's another example. We had an example earlier. N's and H's stuck together. They also form a slight charge when they join together. And there's also an oxygen in there, which probably has a bit of a charge as well. So here are a few more examples of acids then, acids and bases. Acids are negatively charged. And what happened here is this used to be an OH, but the H fell off and became a hydrogen ion, making the liquid more acid. So now it's an O- right there. So if you see a negative there, this is a negatively charged amino acid. If you see a plus here, these are positively charged. These are bases. Okay? Now, when we make a protein, the first part of a protein is known as the primary structure. Primary structure is formed when we form covalent bonds between amino acids. So the backbone is where this occurs. Okay, so peptide bonds form, which is the name of the covalent bond between amino acids. So it's a dehydration reaction. An OH comes off here, hydrogen off here. We make water. We form a new covalent bond. Okay, this is what happens at the ribosome. So what happens is your DNA contains genes, which codes for your messenger RNA. And if you know what codons are, every 3 bases in messenger RNA is a code for which amino acid goes where, and that codes for the protein, the order of the amino acids in the protein. So this thing right here we call a polypeptide, which is just a young protein. It's the order of amino acids in the protein. So there's the 30th amino acid, there's the 125th amino acid down here, and the order of where to put each one of these was coded for in the DNA. The next thing that happens is that secondary structures form, and that's when that long chain of amino acids folds up. In this case, hydrogen bonds form between parts of the backbone, and that's what holds this in place. And this forms what we call secondary structure. So primary structure is the order of amino acids. Secondary structure is formed by hydrogen bonds in the backbone, and it can form things like an alpha helix or a beta pleated sheet, which is like a pleated piece of paper or something. Okay, that's secondary structure. Then you fold these up again, and so now the R groups come into play, and that forms tertiary structure. So an R group might fold away from water or toward water, or an R group might covalently bond with another R group, and that folds the protein into what they actually call a globular protein. But it has a very specific shape. It might be a protein with an active site on it that has a job to do in the body. But not all proteins are done yet. Sometimes you take one of these and then you build another one, so you get another primary structure, fold it into a secondary structure, and fold that into a tertiary structure. And so you have one of these things. Then you make another one, and you stick them together. And you can end up with a quaternary structure, which is more than one. Let me just go tertiary structure. So if you were to look at that, like hemoglobin, for example, has 4 different tertiary structures in it, which means there's 4 different primary structures in it that were folded into secondary structures, folded into tertiary structures, then stuck together. So here's an example of tertiary structures being held together. And so hydrogen bonds between those side groups can form van der Waal interactions, or hydrophobic interactions, which is when things that are hydrophobic turn away from water. Van der Waal interactions are just really weak interactions that form between molecules because of the random motion of electrons creating tiny charges. It's not something I went into much in this course, but it's the same way that the same kind of reaction that allows a lizard to climb up a pane of glass. It's a very, very weak, even weaker than hydrogen bonds, but a very weak interaction. There are covalent bonds that form between sulfur groups, a disulfide bridge, and there are ionic bonds that form between side groups. And this is what holds that tertiary structure in just the right position. If it has the wrong position, it doesn't work. If a mutation changes one of these amino acids to the wrong one, it changes the structure of the protein and it no longer functions as it should. Structure and function are tied together, and this all goes back to your DNA. Your DNA is the code that tells the body where each thing needs to go to get just the right shape out of that protein to do its job. So there's a quaternary structure you can see there. And within that quaternary structure, you can see the color-coded. And there's one tertiary structure, there's another, there's one, and there's another. And if you look at the the tertiary structure, you can see that there's like beta-pleated sheets, and there's an alpha helix in there. You can see the secondary structure as well. Hemoglobin, once again, you can see those curlicues of those alpha helices, the secondary structure. But there's 4 different subunits here, 4 different tertiary structures glued together to make a quaternary structure. And as you know, if your DNA is mutated, you'll get a different shape in that quaternary structure, and that's how you go from having normal hemoglobin and normal red blood cells to sickle cell shaped blood cells, because the hemoglobin has an incorrect shape in the case of someone with full-on sickle cell disease. So I'm going to end with this question and let you answer it, and then we'll pick up with one more lecture video for this chapter.